So welcome to another wonderful Harvard Science Book Talk. I'm Greg Heston. I'm the Associate Director of Science Ed, and I teach physics here. And on behalf of the Harvard Division of Science, the Harvard Library, and the independent Harvard Bookstore, thank you all for supporting it. Welcome to this wonderful book talk by Professor Steve Ramirez. We're going to hear about his book, How to Change a Memory. Before we get to that, just a few little housekeeping things. As always, if you want to know about future events, you want to see videos from past book talks, just look up Harvard Science Book Talks, Google it, you can find our website, look on YouTube, you can find the videos. In a week, we have Nayesh Ashforty in conversation with our own Lisa Randall, talking about the book Battle of the Big Bang, The New Tales of Our Cosmic Origins. In 2 weeks, we have our own John Huth in conversation with our own Jacob Randis. And the book is A Sense of Space, a local's guide to a flat Earth, the edge of the cosmos, and other curious places. And on December 3rd, we have Felice Frankel talking with our own Melissa Franklin. And the book is Phenomenal Moments, Revealing the Hidden Science Around Us. That's beautiful pictures and science, and it's very cool. You can see our little place in the universe. And then I guess these other books are the edge of our universe and the beginning of our universe. Many parts of our universe you will see in our future talks. But we will be exploring our internal identity, our personal universe, in Professor Ramirez's talk. Professor Ramirez is a professor of psychological and brain sciences at Boston University. He is an award-winning neuroscientist, and his TED Talk is amazing. Even after this, you will want to see that too. It's just great. Steve, Professor Ramirez has been on CNN, NPR, BBC. His work has appeared in New York Times, National Geographic, Wired. Okay, I could keep going on and on. When I was at Nova PBS, I was working on a documentary, and I was looking for a top expert, and everyone said, talk to Professor Ramirez. And I spoke to him, and it was one of the most valuable conversations I had. You told me about the really cool work you were doing by changing memories, how you could get rats to just have fear, associate fear with spaces. I think this was your graduate work. They would associate a fear with spaces they didn't previously associate fear with, which is a common graduate school experience. We don't all get a nature paper out of it. Some of us get night terrors. But it's wonderful. It's great to see you again. Great to have you here. And I'll just read one quote. This is Elizabeth Loftus, author of Eyewitness Testimony, which says, he shows us that by modifying memories, we can heal the mind and promote health. We can change our lives as we know it. Rarely will you find a scientific memoir that leads you to say, I couldn't put it down. This is one. We will get to hear all about that. At the end of the talk, you'll have an opportunity to ask your questions. So we'll have the mics going around, wait for the mic to come to you. And then following that, go over to Cabot Library, where Professor Ramirez will be signing books and chat with him. I will remind you, please do that. Go over to the library rather than coming up down here, so we can all join in that reception space and clear the room. And with that, Professor Ramirez.
Thank you. Thank you, guys. Alrighty. Everyone can hear me okay? Awesome. Thanks so much for being here, everyone. It means a lot twofold. Like, one, because we're celebrating science on a Monday, which is pretty amazing. And two, as I'll talk about, it, in a very literal sense, has been a lifelong goal to write a book. And the goal of today is to really unpack why I've always wanted to write a book, the stuff that the book is about, and hopefully today, this talk, if anything, is more of a supplement to the book. So I do appreciate everyone being here, and hopefully the book connects with you in any way that you find meaningful. The best advice I was given was that once the book is out, it no longer belongs to me, it belongs to the world. I really believe that. So I would love to hear your thoughts about it after you're done reading it. So for today, instead of going through the book itself, since it would be nice to have everyone read it, today's more about like how this came to be. So how to write this book, and then my quest to write a book. So like the book itself, I'll weave in some personal stories, some very personal stories, as well as a bunch of science. And that really was my goal in writing this, was to weave the humanity with the amazing discoveries that are possible, that have been possible in contemporary neuroscience, and to weave both, right? Because when I leave the lab, the human doesn't end. Like I'm still me, and when I'm in the lab, I'm doing science. When I leave the lab, it's still me. So the goal here was to really convey that as close to a 360-degree view of the process of doing science and the people, in this case myself, behind that process. So I'll start with just a little bit of contextualizing of myself, and then from here we'll move into my 5-ish years of grad school and some of the several discoveries that we made in that time, and then end with what it really means to change a memory, and would we even want to change a memory, and how we can do that for things like healing, right, and things that enable well-being at the end of the day. So just a brief tour of myself here. I was born in Everett, or I was born here in Everett, Mass, like 6 miles north of Boston. And all my family's from El Salvador. And I have a lot of childhood memories that I go back to time and time again that they do kind of change each time I recall them. I barely remember this picture. I do remember being in love with my Pokemon card collection. This was like seminal for me as a kid. My dad still has our card collection locked away safely at home. This is my cherished firstborn here. So okay, growing up, I really enjoyed Pokemon cards. This does become important in a second. And then this is me a little bit older, just going back to visit some family in El Salvador. Throughout this time, I always liked the humanities, the arts. I grew up playing music. I really liked science because of shows like House and Scrubs and so on. I liked the idea of medicine, and I was pre-med for like a week, and then that ended very quickly. But it's all to say that I really had no idea what I wanted to do with my life, but I did know that I just like exploring every topic ever. So growing up, this is my childhood home in Everett, 66A Malden Street. So when I was a kid here, I would spend my free time either collecting more Pokemon cards or going to the bookstore just to see like what's the latest on all the things that I liked. The arts, the humanities, medicine, music, the Da Vinci Code, A Brief History of Time, just everything under the sun that I thought, Animorphs, like anything, I'm like, this is cool. Like, this is cool. It's really amazing that someone can know so much to write a book. Like, as a kid, that idea kind of enchanted me because it's kind of wild. I thought, like, how can someone talk for 300 pages? Like, that's a lot of knowledge that in one person's brain that now is put in a bookstore. And as a kid, I was just in love with the idea that, like, if you're in a bookstore, it's because you did something important and you did something that maybe matters or is of value to humanity. And I liked that idea as a kid because, I don't know, I just wanted to feel important, right? And that was one way that I latched onto that dream for a pretty long time. And it really came to be in the Barnes & Noble that was in Route 1 in Saugus, where I'd go there on the weekends and spend just as much time as possible seeing just what's new in the physics section, what's new in the history section, and to really just get a glimpse of all that humanity knows. And I think that to me was the most enchanting idea of all, was that you're just surrounded by the wealth of knowledge that humanity has accrued, and you can pick it out in any book and dive into it and find out everything that there is to know about whatever it is that you want to know about. That was cool. So I held on to that idea for a while, and I went through college kind of undecided. I didn't really know exactly what I wanted to do, and then it wasn't until my junior year at Boston University that neuroscience was like invented as a major. That was the year that it came to be. And through like a series of serendipitous events, I joined a lab that happened to study memory. And I'm convinced that if the lab studied like algae, that's what I would be studying today. Because like, no offense to algae, because it's really cool, but I'm convinced that I would have done with my life whatever that lab was doing because I loved the people that were working there. Like, I felt like I could raise my hand in lab meeting and ask whatever stupid question I wanted to ask, and I didn't actually walk away feeling inadequate. I walked away feeling like that's amazing that we can ask questions as a junior in college that the field of neuroscience doesn't yet have an answer to. So I applied to grad school mainly because my mentor at the time would have been mad at me if I didn't apply to grad school. And I thought, well, I am in like with science and not necessarily in love with it, but I'm in like with it enough to keep going back to the buffet of ideas and seeing if I fall in love with it over time. So I applied to grad school. This was finals week of senior year in the fall, or in the winter, where I remember I was in the dining hall, and this is the first memory to unpack for this talk. I was in the dining hall, and I had like a colossal bout of food poisoning, and I remember vividly, I was eating a mix of Lucky Charms and Cap'n Crunch, because those are the only two things I could hold down, and then I check my phone, and I get this email. And this was when I got into grad school, so super exciting, very like unreal. I actually thought my roommates were playing a prank on me because I wasn't expecting to get into MIT, let alone get an acceptance letter before, I think this was even before Christmas. So I get this email, really excited. I pick up my phone to go call my parents just to yell like, this happened, right? So I call my dad, and I'm like, Dad. And then as soon as I say Dad, I drop my phone in my bowl of cereal. And like, this was before phones were milk-proof, so my phone was dead, so I had to run back to the dorm and call my parents from another phone and convince them that I hadn't been kidnapped, because the last thing they heard was Dad, and the line cut. So I was not in fact kidnapped, so I needed to convey to them how excited I was, and it was very much a full circle moment, right? That I get to continue my education in neuroscience and dedicate the next 5 years to finding out something super specific about how memory works. So, okay. At the time, I knew that there were a handful of ideas that I would love to write about, but I had no idea what the topic of a book would even be. So I put it in the back burner, like just in the back of my head. Grad school does a great job at keeping you busy anyways. So I joined Susumu Tonegawa's lab. This was the lab at the time, and it was really an amazing time to be able to just do kind of like whatever experiments on neuroscience you really want to do. Like, it really was a playground for science. And the thing that made me join the lab more than anything, like my previous lab, was the people. That the people there were like, it was great, right? Like, if something works, you go across the street to the bar to celebrate afterwards. And if it doesn't work, you go across the street to the bar afterwards to commiserate. And it goes a long way to have that kind of support structure, right, in any environment, really, but in the lab in particular. And that's the thing that drew me there. So the core of the book, as well as just the half a decade of grad school, was my friendship with my late friend here and lab partner, Shu Lu. So when I joined the Tonegawa lab, I spent the first couple of months reading up a lot of literature on epigenetics, which was not at all what I would go on to study. And then one day, I went to Susumu's office just to ask him for a bit of like guidance and some help on what projects to embark on. So he was like, you talk a lot and you smile a lot, and I know someone who does that as well. And he introduced me to Shu, and we immediately hit it off as lab partners, because we both had a very, at the time, probably naive but wide-eyed view of what science at its best could be by partnering up. Like, we were I remember literally the first time we had a conversation about science, we were at our building's happy hour, and after a couple of drinks, we're in the elevator, and we were like, so okay, obviously we work well together. Let's just be co-first authors. So like, we share credit for all of our papers and give talks together, give posters together. Let's kind of try to dynamic duo it out, because two brains are better than one, and science becomes way more fun when you actually have someone to share all of the successes with. So we immediately partnered up, and it was fantastic, because from that point on, he introduced me to this idea of what we called Project X that I unpack in the book. And it was called Project X because the lab had spent the previous 10 years trying to develop the tools needed to execute on this idea of trying to artificially activate a memory. And the joke was that it was Project X, and to try to like unjinx it so that it wouldn't last another 10 years. So we joined forces here to try to test an idea that had been around really since the 1920s, that there is a physical copy of a memory in the brain somewhere, and that we could somehow flicker on the cells that are involved in housing that memory to then force the recollection of that memory. That was kind of the working hypothesis that we started off with here when we worked together. The area of the brain that we decided to dive into was one that has a rich history in being involved in all sorts of memories that we hold near and dear. It's called episodic memory. They're the episodes of our lives, the memories that we make and that we hold near and dear and that we usually mentally time travel back to when we're sharing a story, like today. I really only am showing you this. This is my hippocampus. I actually got to get an MRI taken for these purposes. Very exciting to actually see that there was a hippocampus and that you have, it's kind of scary going under an MRI for the first time because you're like, they're just going to see a black hole and then that's kind of weird, but like, you have two hippocampi above your ears tucked inwardly, and there was like 70 years of research that gave us good reason to believe that if there was anywhere in the brain that we could jumpstart a memory, it's probably in the hippocampus. It's a little biased because the field happened to study the hippocampus for 70 plus years, but the tools that the lab was developing were kind of perfected for the hippocampus especially. So we thought, great. There's like a suite of genetic engineering tools that we can use to try to identify cells that hold on to one memory. We can get them to glow a particular color or to be artificially activatable. But let's test it out and see if this actually works. Like, can we artificially activate a memory in the brain or not? We partnered up, and Shu and I decided, like, okay, we're going to do these experiments together. We're going to share the work to the world together. Like, this is us giving our TED Talk in Faneuil Hall in Boston. And we thought, if we could at least convey this sense of togetherness to the field, then it doesn't have to be like a one-person show, right? Like our ideal was kind of like, you know the videos of like when the rover lands on Mars and you see all of the control room in NASA going crazy that the thing landed? If that could happen in neuroscience, then that would be fantastic because you have a room full of people dedicated to a common cause, such as trying to restore health and well-being to the brain, right? So we decided we're going to work in the hippocampus. We want to try to artificially activate a memory. And then the tools to be able to do this were particularly perfected for rodent models and mice in particular. So this was the first mouse that we ended up using to test this idea of can we artificially activate a memory. And I'll spare a lot of the nitty-gritty genetic details, but the idea here is that you can find the cells that hold on to an individual memory in the hippocampus. You can genetically engineer just those cells to respond to brief pulses of light, like this laser. This laser is actually strong enough to activate, it's enough milliwatts that come out to activate those brain cells. So brain cells normally don't respond to light. It's just kind of a black box in there. We genetically engineer these cells to respond to light through a technique that you may have heard of called optogenetics, like opto meaning light and genetics meaning genetics. So the idea is use optogenetics to try to photostimulate memory-bearing cells to activate a particular memory. And to give you an example of scale here for anyone that doesn't do rodent work, on the left is an intact mouse brain, and on the right is a chickpea. So that's the size between these two. Yeah, I know, this slide like ruined salads for me because I can't look at this the same anymore. So that's the scale that we're working on here. The very first positive experiment that we had that produced positive results is one that I show now in basically all of my talks because what we saw was cells in the hippocampus that processed a particular fear memory in this case, and I'll get to why fear in a second, and that we could find those cells in the hippocampus and trick them to glow green so that we can visualize them under a microscope, which are the green cells that you see here. And this is a cross-section of the hippocampus that's like 50 microns thick, so it's thinner than a credit card in this case. And what we can do is go in, find the cells, get them to glow green, and we can photo-activate them with a laser here to see if activating those cells activates the memory, right? That was the goal. A brief aside here is that when we were doing these experiments, the one that ended up working was the one where we did surgery on 5 animals. In 4 out of the 5, we didn't see anything. In the 5th animal, we saw evidence of memory recall, and when we went to go see what made that 5th animal different, we had actually botched the surgery, and we were aiming somewhere up here, and we went down like 150 microns deeper and hit this part of the hippocampus, and that's where all the magic happened. So I like to tell this story because it's an example of like, It's both. It's hard work and good luck that leads to a discovery, right? Like it's going to be a combination of both. And we had put in the hard work to get these animals ready and to do the surgeries on them. The luck part was missing by 150 microns. And in that space is the difference between a negative finding or literally the rest of your career. So that mouse was a big deal because it was our first bit of evidence that maybe this is actually working. So the way that experiment goes, and this is both the opening of the book as well as the opening of chapter 1, because I revisit this memory several times the way that we revisit it here. So okay, the way this experiment goes is, imagine that you put an animal in what we'll just call box B. We give the animal a fear memory, and we study fear in particular because it's a pretty straightforward readout in animals, and it has pretty direct implications for PTSD and anxiety in particular. So what we did here is we give the animals a couple of mild foot shocks. It's more like static, right? It doesn't hurt the animal. It's like when you get static and surprised. We find the cells that hold on to that experience of the animals being in box B. They get these mild aversive stimuli. The cells in the hippocampus that were glowing green, those are the ones that are active now that are holding on to this memory. And we've tricked just those cells to respond to pulses of light. So those are the ones that we can photoactivate. The $1 million experiment here is we put the animals in a completely different environment, so different sights and sounds and smells and floor and textures and so on. And what we do is, first, the animal has no reason to be fearful of this environment because nothing negative has technically happened here. But if we go in and photoactivate the green cells from the previous slide, if we go in and photoactivate just those cells, does the animal behave as though it's fearful even though it's in a safe environment? And the way we measure that is through what we call freezing behavior. And it's called freezing as the name kind of gives it away. If you were to hear a loud noise right now, most of us would kind of like tense up and freeze. And then fight or flight and so on would kick in. So it's an evolutionarily conserved response that we share with rodents. So the opening of the book is this day, where we are in lab. This is the 5th animal, and we were kind of like, we weren't really that bummed because science doesn't work like 99% of the time. So when the first 4 animals showed nothing, we were like, I mean, this experiment had every reason not to work, so it's very okay. Now, this video is going to be split into 2 parts. The first part is important because it's a control for light. And what I mean by that is you could imagine that if someone put an optic fiber into your brain and started shooting lasers into it, you might freak out. So we want to do that without activating anything in the brain just to see if that changes the animal's behavior or not. So we're controlling for the effects of light in that sense. So you can ignore the caption. This animal is in the safe environment. It has no reason to be scared of it. It does not have a fear memory yet or anything at all. We're just shooting light into the brain to presumably do nothing to the brain. And as you can see, the animal's behavior doesn't really change. That's great, because that means this control works. It didn't freak the animal out, right, to shoot light into the brain. This is the same animal. The animal has a fear memory, and the cells that hold on to that fear memory are photoactivatable. And when we activate them, the animal immediately starts freezing. That is, like in 15 seconds, that's kind of like my career. And that day, we were like, this is the opening of the book, and that day we were like, okay, it's an N of 1, so we got to do this like 1 billion times over again to make sure that it's real. But I think that there's something real there, like as the saying goes, right? If it's an N of 1, like if you have a talking dog, you don't need an N of 2, like the dog is talking, like that's proof enough, right? So, but no, in science, obviously, we got to reproduce this and do it again and again. So we did that. We did a bunch of controls. This paper, it's the first paper that we published together, and it's my favorite, not just because Shu and I were co-first authors on this, as indicated by the asterisks, but because if you read this paper, it's one finding. Like, it's one finding in figure 3A that shows that we can photoactivate a memory. Everything else is controls, just controls and negative data. And I don't know, I kind of was enamored with that idea that we have one finding and then we try to break it from every angle. And thankfully, it's survived a decade and a half now. But I like that because to me, it was, it felt, I don't know, elegant, right, for lack of a better word, that we could have just one straightforward finding. It's a seesaw shape of turning a memory on or off or on or off. Everything else in the paper is controls to try to show that we're doing what we think we're doing. So when you publish a paper that claims to reactivate a memory, the public tends to listen. The kinds of comments that we got afterwards were pretty great. Like, we'd go on different forums and yeah, it would be like, oh my God, finally so much more to come, rewriting of memories, mental illnesses, the future is awesome. This was in 2012, so not really prescient, but that's, the future is awesome for the field. There was, there were other comments on like, we should make a movie about this and call it Inception. That's a great idea, a fantastic idea. And at the time, this now begins a little bit of chapter 3 and 4. At the time, Inception had already come out, and I would listen to the soundtrack nonstop. I was kind of like the main character syndrome of just my own movie here, listening to Inception while working on a project about Inception. And I thought, okay, we can reactivate a memory, but can we change the contents of a memory now? Can we try to warp a memory and kind of pull off a sort of Inception-like feat in this case? And I can give you every hypothesis for why that's important and why we want to know about the malleability of memories. But Shu and I were just like, this is just kind of cool, and I think it has pretty profound implications for memories. So at this time, I remember taking some mental notes of, whenever I write a book, I would love to write about the malleability of memory, but in particular, false memories. It was probably the topic that I was most passionate about in grad school. So when my wife and I would make this joke where it's like we would leave work to go work. So when I would leave work, I would go work at, this was Eastern Standard in Kenmore Square when it used to be there. And it's like a few blocks away now. I would go here basically every day after work and sit in this, I don't know if you can see this well, but I'd go there and sit in this corner over here, order a couple of drinks, and just write down ideas of like, it would be great to talk about this aspect of false memory, or maybe in the intro for our paper to be, I can intro this aspect of memory to connect our findings. So I just started writing a bunch of bullet points of ideas that I would love to write about one day. And that is the seeds of chapter 4, was over a couple of years actually at Eastern Standard, over like martinis, false memories, trying to combine those somehow into a commentary on the malleability of memory. So the malleability of memory was something that I thought was particularly interesting and is the focus of a big chunk of the book, hence the title, Change of Memory. And it's something that we all experience it without maybe even knowing it. Who's heard of the Mandela effect, for example? So like a pretty good amount of people. For example, or to give an example of more sort of like pop culture-y memories that we all have that we think are true that might not actually be true and are relatively inconsequential. All right, for peanut butter, is it Jiffy or Jif? Great, it is Jif. Jiffy is Skippy plus Jif, so that's good. I was maybe like 80% got it right. Okay, a little bit tougher. The Monopoly guy, does he have a monocle or not have a monocle? That was a little more 50-50, right? Like that. So the Monopoly man, I almost called him the monocle man. The Monopoly man does not, in fact, have a monocle. What I think is happening is that we're taking the monocle from the Planters peanut guy and then copy and pasting that onto, right, but then like that is a perfect demonstration of how silly putty-esque our memories can be. Okay, Febreze, is it 2 E's or 1 E? All right, everyone who does not clean their house just told on themselves. Like so Febreze is 1 E. Pikachu, does Pikachu have a bit of black on his tail or not? Yes. Pikachu does not, in fact, have this on Pikachu's tail. Yeah. You can fact check me in real time. Pikachu does not have a black mark on his tail. Kit Kat, is there a hyphen or no hyphen? No hyphen. Okay, everyone eats Kit Kats. No hyphen, great. Last but not least, Oreos, double stuffed with 2 F's or with 1? I'm not gonna lie, I forget, but I'm pretty sure it's the 1. So yeah, the Mandela effect is just this popular example of the things that we misremember occasionally, and it happens to all of us, right? Like in movies, this happens. Like some of the more classic examples are like in Snow White, the evil witch never says mirror, mirror on the wall, it's magic mirror on the wall. Or in Star Wars, like Darth Vader never says, Luke, I am your father, spoiler alert. It's more, Darth Vader says, no, I am your father. So it's subtle, but it's different enough that it's been kind of like saved as in our collective memory as the version that it is not. And I think that perfectly encapsulates what memory actually is. So there's a bit of irony here, because when you look in the human brain as we're recalling something that isn't, so as we're recalling a false memory, the area that, or an area that becomes very much active is the same area that is involved in encoding all of our experience and episodes of our past, namely the hippocampus. So this is an MRI signal, a cross-section of the brain, hippocampus activity here and here in red. The important line here is the orange one, that when you recall something that's true, there's a big increase in hippocampal activity. When you recall something that's false, there's also a big increase in hippocampal activity. So this begs the question, right? Like, why does the area that helps you make the memories that you hold near and dear, why is that area also active for memories that are not true or that are not like veridical copies of the past? It's a good question. This gets tackled in both chapters 4 and towards the end of the book, but there's a lot of cognitive feats that being able to have malleable memories is thought to enable. So the quick spoiler, but it's not really a spoiler because we unpack it a lot more, is that by being able to recombine elements of your past, the theory is that our brain is constantly actually making predictions of the future, and by using memories as building blocks, we can combine and recombine them while we're awake to imagine a future. So when we imagine a future, we import our previous memories into some weird kaleidoscope that helps us imagine an event that hasn't happened yet. And you all have probably experienced this most intimately while dreaming. A hypothesis around what dreaming is those building blocks being combined and recombined in real time. So false memories were cool. And that's something that at the time, Shu and I thought, let's take this seriously. And let's try to see if we can dive into the hippocampus once again and see if we can alter the contents of a memory by tinkering around with these memory-bearing cells in the hippocampus. So this was in the winter of 2012, right after our first paper on activating memories had come out. Susumu invited Shu and I to come to New York because he was invited to give a talk at Columbia. And he was like, you guys just published a big paper, that's exciting. All expense paid trip to New York, awesome. So let's go and we'll watch the talk and then we'll go out and celebrate together. So Shu and I went out and we bar hopped around Times Square for the entire night. And then we ended up at this was 3 a.m. at an Irish pub in Times Square where we were like, okay, let's take it seriously now that maybe we can implant a false memory. How would we do it? So as one does at 3 a.m. in Times Square. So we thought, what would be the simplest way of designing an experiment that might be able to provide some proof of concept of warping the contents of a memory? So we got a napkin and we wrote down this idea of box A, box B, box A. This was the simplest way that we could try to alter the contents of a memory. And the way this experiment goes, and this is the schedule that we ended up adopting for this experiment, is we're once again back in our environments, but we'll call this one box A, right? So box A is just a neutral environment. Nothing fancy is happening here, no shock, no aversive stimuli or anything. But the cells that are active in box A, in the hippocampus, are the ones that now are we're making photosensitive, or we're using optogenetics to make them light sensitive. In other words, we have optical leverage now over the memory of box A. If we shoot light into the brain, we should, in theory, be able to activate the memory of box A. The next day, we put the animals in box B. This one has the grid floors that administer the mild foot shocks. And now the experiment that we do here is we say, while the animals are getting these foot shocks, what if at those moments we reactivate with light the memory of box A? Because that was the attempt to artificially link the memory of box A with the real aversive stimuli that are happening in box B. Because if that works, then when the animals are placed back in box A, they should show fearful behavior in box A, even though technically nothing negative happened in box A. Does that kind of make sense? Great. So to set the stage, this one was Christmas Eve of 2012, and I remember this because we had a rock, paper, scissors game over who had to go into lab on Christmas Eve, and I lost. And I went in, and I remember because my parents were both waiting outside of the Brain and Cognitive Science building, and I told them, this experiment is only an hour, but it happens to be probably the most important hour of both my life and this experiment. And then my brother and sister were waiting home for holiday dinner, and I figured, the hour can be okay to wait here. So I go into our behavioral testing rooms, and we had a handful of animals now that were ready for this particular day here to test whether or not the animals were in fact recalling what we called a false memory. Because again, they should freeze here even though the negative event happened in B. So to show you what this looks like, we're back here in box A. So this is when the animals are in the neutral environment. Yep, they're in the neutral environment. The cells that are active during the exploration of this neutral environment are the ones that now become light sensitive. So that's what's happening in this period here. So now we give the animals a couple of mild foot shocks while we artificially activate or photoactivate the memory of box A, which is what's happening here. That was December 23rd. This is December 24th, where we put the animals back in the neutral environment, and all of them showed this kind of behavior. So I shot off an email to Shu being like, holy crap, I think this really worked. And I sent him a PDF that was just a single graph, and it was inception.pdf that I sent him being like, I think this worked. And I went to Susumu's office and showed him the graph too. And typically, this was a PI lesson that has stuck with me forever from a mentor. I brought him the graph because I remember thinking like, I know that this worked, and that's kind of amazing. And he shook my hand, and he was like, that's so exciting. He's like, what's your next project? I was like, what do you mean what's my next project? This is like, we're not even done with this one, and we got to publish this one. And he was like, I learned there that you obviously have to have a vision and be like 10 years ahead in the field, which I respect. So we published this paper, and this was really exciting because we had shown that we could artificially link this neutral memory with aversive stimuli. And now the floodgates really opened because we could activate either a neutral memory or a fearful memory, or what I'll talk about in a sec with positive memories. And the way that we ended up doing these experiments and publishing these experiments is the topic unpacked here in chapter 4. And at the time, I was elated, right? Because the deal that Shu and I made that first day that we really got to know each other in Buildings Happy Hour, this came to be where we were co-first authors for our first paper, and now we were co-first authors for our second paper, and it was a second paper in just as many years. So we were excited that there was this burgeoning field of memory manipulation that we were smack in the middle of now. And it was really a privilege to be able to share co-first authorship, switch our names, and then we're like, damn, like this, it turns out this whole teamwork thing is a real thing that really works, so let's continue doing it. And so we did, and our team continued to grow. And we, again, like main character syndrome here, we called ourselves Team X because of Project X. And hence the X gang signs here that we're throwing up, like where our team began to grow, and it produced pretty amazing results pretty quickly. So for instance, one postdoc in the lab, Tomas Ryan, discovered that you could pull a memory out of amnesia, so show that you can restore a memory that was thought to be lost in cases of amnesia. Roger Redondo went on to show that you could switch a negative memory, you could switch the cells that hold on to a negative memory to hold on to a positive memory, and vice versa, that the cells that hold on to a positive memory, you can switch to hold on to a negative memory. That is all now the topic of chapter 5, which is to try to change memories in either a therapeutic manner or in a manner that just tells us more about the basic science of how memory works. So after that idea, I thought, okay, plenty more to talk about in terms of memory's malleability. So after work, I would, once again, go back to work, and I would go, in this case, to the Trident bookstore in Newberry Street. And this is where I would sit in this corner over here by the pillar and just write down ideas on memory malleability and basically write down the seeds of what would become chapters 4 and 5. And then at the time, right, I would go there, once again, order a couple of drinks, write ideas for chapter 4 and 5, try to muscle through it all, and then keep those ideas in the back burner. So the last project that Shu and I worked on, and this kind of, this will end a little bit of the more science part before going into more like sort of the personal journey of writing a book part. The last part, we thought we could activate a negative memory, that was 2012. We could create a false memory, that was 2013. So the natural question now is, what about positive memories? And that one was particularly meaningful because we thought, could we transform the psychological construct or idea of memory into a very real biological antidote? Like, can you look at memory as not just something that the brain does, but something that you can tinker with in some kind of therapeutic manner? And positive memories seemed like a natural place to start. And we all have these, right? More than most like antidepressants and anxiolytics, like things like mindfulness or gratitude journaling are like parts of silver bullets for good health for our own just cognitive well-being. But it takes effort, right? It's like a workout for your brain where you really have to sit with the positive experience and be intentional about re-experiencing the positive experience, like with gratitude journaling, to really to get the benefits of it. Now, at the time, I used to hate public speaking literally more than anything else because it was a disaster. It was just like the #1 anxiety-inducing thing that I could possibly think of, besides like a first date or something. Like it was awful. So what I would do before public speaking, and this was particularly in lab meetings in grad school, what I would do is I would hype myself up by revisiting the positive memories that actually meant something to me. Born and raised in Boston, so this was obviously the Pats Super Bowls. So I would actually go back and rewatch the clips of the Pats winning the Super Bowl, so that I could get a little bit more excited and my heart rate could go a little bit more up, so that then my body could match the sort of roller coaster of thoughts that I was having before a talk. So there's some theories of emotion here of aligning sort of your thoughts and your body to try to be on an equal playing field as well. This was my pump me up speech, right? This is the way of looking in the mirror and saying, you got this, you can go give this talk. You're not gonna keel over, it's okay, kind of thing. So they're really potent, and there's a lot of science behind this. In humans, for instance, it's been shown that if you really actively recall positive memories, your heart rate goes back to normal, your heart rate variability tends to be a bit healthier, your blood stress hormone levels go back to a baseline faster than as if it was a negative memory or a neutral memory. Positive memories activate all of the brain's reward and dopaminergic circuitry and so on and so forth. So the things that positive memories do profoundly change the rest of your biology, and we really wanted to take this idea seriously of like, how does it do that and why does it do that? So in mice, there's a test that we could use called the sucrose preference test, which is basically like humans, mice tend to prefer sugar water over regular water, the same way that most of us would prefer something with flavor than just water, for example, soda compared to water, let's say. So healthy mice prefer sugar water about 80% of the time relative to mice that we think show symptoms related to depression and anxiety, where they show a 50-50 preference, which is another way of saying that they don't show a preference at all, right? We call this anhedonia, an meaning no or lack of or without, and hedonia meaning more the ability to seek out or experience pleasure. So this is a way of modeling anhedonia in animals, and it's one of the core symptoms of depression. So we asked, if we have animals that show this anhedonic-like behavior, can we reactivate positive memories to restore their preference for the sugar water? So the positive memory here for mice, there's plenty. Cheese is actually one of them, like that they do like cheese. The thing that we use that's the most potent nowadays is like a mix of cheddar goldfish and condensed milk. Like they actually fight each other for it. And there's also social rewards, right? The animals socializing with each other. And as well as cocaine and sex and all of the stuff that we've just known forever that animals will find rewarding. So we use social reward in this case as our positive memory. And what we found, the important line here is just the orange, is that when the animals do not have a positive memory activated, when they show this anhedonic symptom, they show about a 50-50 preference for sugar water or sucrose preference. And this is when the light is off. But when we reactivate the positive memory, this is the orange line here, it goes up almost to the same baseline as animals that are not stressed. So in other words, reactivating a positive memory reverts the animal's behavior back to how healthy animals are behaving in this test. And that led to our last paper together. And this leads to the end of chapter 5 and then part 2 of the book. Because at the time between finishing that project and publishing this paper, we finished that project around January in 2015. And then in June in 2015 is when the paper came out. In that time is when Shu unexpectedly passed away. So that was hard, to say the least. And that was the part where what memory means to me just became like terrifying and magical all at the same time. So anyone that's lost anybody knows how much like the realities of grief are just this thing that's so individual and so personal. And I think that there's a lot of ways of confronting that has everything to do with memory, which will be the last couple of minutes of this talk. So just as an example of how a memory can change here, I get really excited, of course, when I see that, oh my God, we published a paper on reactivating positive memories to curb depression-related symptoms. What I see here is Shu and I, we couldn't be co-first author for this paper again. And I learned that when someone passes, what a journal does is they put up these two crosses next to the person's name. So when I see this paper, I think about the memories that we have together, but I can't not see the absence of the person as indicated by the two crosses. And that memory has changed in the last decade, and this is unpacked a lot in part 2 of the book. It's changed a lot because it goes from impossible to handle to slowly trying to make sense of that which we can't really make sense of, to what does this mean to me and how can I find some kind of purpose as a person to help let someone into your life but in memory form now, right? So the morning that Shu passed, I remember walking to work and I called my dad, and I remember thinking like, in that moment, I remember just thinking like, this was the sunrise of that morning. And I remember thinking like, okay, I don't know if it's just romanticizing the moment or not, or maybe just there's a difference between experiencing something or recalling something. But I remember thinking, this is a beautiful sunset, and maybe this is weirdly the universe's or my head's way or some way of just telling me like, let the light in and be awake for this moment, right? Like it'd probably be the hardest moment you gotta go through, be awake for it and be present for it. So I learned from there just how much making new memories can really be a way of addressing something as like ephemeral and intangible as grief. And I stumbled on this cartoon, which is like I think this is like one of the most profound cartoons I've ever seen, and it's my favorite one for making sense of losing somebody, because the reality is that everyone is destined to become a memory, right? That's the price that we pay for having the luxury of having a life, right? So grief was something that I learned was full of all sorts of memories, good and bad, that you're recalling throughout all of the stages of grief. But the reality, both brutal and kind of beautiful, is that it never goes away. Like, grief isn't something that you get over. Like, you don't. You carry it with you like a 20-pound dumbbell on your back that you get stronger, or however we want to call it, to learn to carry that with us for the rest of our lives. The kind of, it's weird saying silver lining given the circumstances, but kind of the silver lining of this all in retrospect is that the way of addressing something as deeply human as grief, I think, is to make more memories, right? And then what you're seeing here is like, yeah, grief doesn't go away, but the way that we live our lives afterwards and the memories that we make afterwards, it grows around the grief, so that the grief, in a sense, is something that we can look at with a slightly different lens. Around that time is when a book agent had reached out asking, would you like to write a book? And it kind of all clicked, because that was the moment where I was like, I now know what I want to write about, and I know exactly what I want to write about, and it has to do everything with memory, as well as the memory of Shu, as well as our time together, and have that be a vehicle for teaching neuroscience, but also teaching, again, a bit of the humanity that underpins all of neuroscience. So at that time, not surprisingly, right, like I am trying to write this ideas of a book at Eastern Standard or at Trident, drink after drink. My relationship with drinking went from 0 to abysmal pretty quickly. And that's the part where I learned that, in retrospect, it makes sense, right? Because the long-term solution for addressing something like grief, I would learn, was writing, hence this book. The short-term solution is the analgesia, the pressing of the button that a drink can really help out with in that moment. Obviously not sustainable, and obviously the kind of thing that leads to pretty detrimental behaviors over time. So I went to New York, and it was in one day. This was now, we're flash forwarding to 2016. I interviewed with like 15 different places in one day to try to like just sell this idea of the book. Landed a handful of deals, thankfully. And then what I would learn in the last few minutes here was how important it was, in retrospect, that the way that I sustainably addressed something like losing somebody was by honoring it, hence the book. And that, to me, was probably one of the most important life lessons that I learned, because I got a handful of deals and I felt like, oh my God, someone actually wants to hear this story now, and it's more than just like a fancy Nature paper. It's just a real story of a real set of people doing real science. So I got the offers for this book, and I sent it immediately to my dad, who wrote an email that I think perfectly captured how I felt in the moment, which was that, congrats, and I know that you deserve it all. Or please know that you deserve it all. And I think that that was the first moment where I felt like I actually feel like it wasn't something that I did with Shu or something that I did in the Tonagawa lab or something that I did with mentors. It was something that for the most part I felt like I did. And that meant a lot to me, to have that kind of agency over like, I can embark on this next stage of trying to honor a friendship and write about memory and teach neuroscience, which is like my big passion in life, all in one. So at the time, that was 2017. Flash forward all the way to 2021. That's 2017 to 2021. I had to address a couple of things. There's a saying that's like you either give up drinking and you get to keep everything, or you give up everything and you get to keep drinking, right? So that was the real choice that I had to make. And this was a journal that I was doing at the time when I was trying to moderate to try to see if I could go from a lot to a little to ultimately to zero. And then I have this here in particular because a couple of friends who are here today sat me down a couple of times and they were just like, in a few words, like, we would like to see you alive rather than not alive kind of thing. And that meant a lot, right? Like that was the kind of thing that I immediately, by immediately I mean after several years, joined a community called Recovery Elevator or Cafe RE. And then that was like the thing that kind of went from zero to health, because I immediately thought like, wow, like you're sitting in a room and the common denominator is that everyone is sharing their memories. Everyone is talking about like their experience with some kind of awfulness that they've had to experience and sharing it. And in that moment, that was the thing that I was like, wow, you're in a room and without really saying anything, everyone gets it. And memory was that thread that brought everyone together in that room to try to address something that they were going through, right? So the thing that I was proud of now years later was that I could say this. This was an interview that I did. And to really go and try to unpack my own memories, good and bad, to try to connect and to try to heal. That led to probably my favorite quote in all of this was that the opposite of addiction is connection. I felt how true that was, not just by going through these meetings, but by really just letting myself share what I thought was once impossible to share, right? So amazing things happen when that kind of connection happens in real life, both in the brain, which is the topic of part 2 of the book, as well as in real life from like a social setting and connecting with everybody. Because now I went from celebrating 10 days to 50 days to then 100 days. And then my wife made me my favorite cake, which was an Oreo cake to celebrate 100 days. 100 days of sobriety at the time was impossible. Like it was insane to make it to that moment. And then I kept going at it, thankfully, and these are the kind of zeros that are worth racking up in life besides zeros after like your paycheck. And so I kept going, and then made it to my 1 year, celebrated with waffles, obviously. Made it to my 2nd year, celebrated in New York with my wife yet again. Made it to the 3rd year, and then around that time got to celebrate my niece's quinceañera. And like I am convinced that I just, not to over-dramatize it, because I don't think it's possible right now, but I just would not have made it to any of these moments if it weren't for leveraging the adaptability that the brain can have when you actually address grief in a sustainable way or in a healthy way. And then it keeps going, right? Because the magic of memories that you can make, the cartoon, right, life growing around grief, the magic that can happen, that can lead to is pretty amazing. Like you go from, this was Valentine's Day and 4th year of sobriety here, and my wife made this just amazing setup to celebrate life together. We got married. Also, just like a really exciting life milestone that we could do that if I was not in the proper like physical or headspace, that memory does not happen, right? Same thing now with the book. Like I got to get my parents into the recording studio to record a few special lines in the audiobook, which comes out tomorrow. And that, that meant a lot to me, as well as finally being able to grow our lives and add to our family and to get our pet Maple, which is the most recent addition to the family. So this is all here just to show the kinds of memories that I think can expand around grief. And they're covered in part 2 of the book, but I think it's just profoundly human to be able to address it. It's not dentate gyrus and the hippocampus and channel rhodopsin and genetic engineering. It's people connecting, right? And then being able to help each other and pull each other out of a rut. So palate cleanser. This is our new dog, Maple. Sorry that this looks like I took it like on an iPhone 3, but it just... But yeah, so to end, the reason I bring those things up is because something like addiction is pretty horrible, but it doesn't have to be it can be, it certainly can be, and everyone's experience is different, but it doesn't have to be, I didn't want it to be my own like sob story. And the reason why I bring those stories up is because I could not have finished the book if I did not grow into the person that I was today, like almost 5 years sober. Like there was no ending to the book. Like I did not know how to end it until I grew into the person that I am now, and I knew how to end it then because I had lived through the way that I wanted to end it. And that, without revealing too much, that to me underpinned in all caps letters the power of what memory can really be and what it can really do. So nowadays, for instance, like when I think about Shu, it's still hard, right? That grief part is still there. Like again, it's a forever thing. But I think of like all that we did together and all that we can still do together by holding someone near and dear in memory form. And it ends up being like somewhat providential. It ends up being like a little bit of a guiding force or a center that you have now, right? And that I think is the way that a person can really live on. And this is the last part here, that the way to really showcase the power of changing a memory. Like changing doesn't necessarily mean undoing, or changing doesn't mean that it has to mean anything less or more. It just means different. So this is just kind of like sort of like, speaking of center, it's more like of a moral compass that I use, and it was something that I couldn't look at before, and now I can not only look at it, but it's just like something that really helps me navigate my way through life, which was the last text that Shu sent me, because at the time, this was 2 days before he passed, we got an invitation to give a talk at the NIH together, and I sent him a text being like, holy crap, like we got an invitation to do a talk at the NIH together, are you interested? And his last text was so simple, but so profound, which just, let's do this together. Like, so straightforward and just encapsulates everything about the kind of dynamic we had and the kind of person that Shu was, and just my overall philosophy and like moving forward in life and in the world, that sense of togetherness, I think, is the path forward as opposed to like isolationism and so on and so forth. So it means a lot. I hope that the book and the talk in this case connects with anyone in any way that you find meaningful. And the preview here is like, just to end, I'll end on the most important page of the book, which is the first page, which is the dedication page, which is everything that we talked about here is both for Shu, and now it's out in the real world and out of my hands and just there to connect with everyone else in a way that, again, is hopefully meaningful. So thank you all for your attention, and I'm really excited for both Q&A and everything afterwards, so thank you.
So I think we have time for a few questions, and then we'll go over to the library.
I'm sure you've probably thought about what the implications might be for someone with PTSD. Could you speak to that?
Yeah, I think of our work as like, just to use a quick analogy, I think of working with mice like working with a tricycle, and working with the human brain like working with a Lamborghini, where we got to ask the right questions to be able to extract something about the principles of motion and momentum and so on and so forth with a tricycle that might apply to the Lamborghini. But if we start asking questions about like motors and carburetors, we're not going to translate anything from the tricycle to the Lamborghini. So the hope is that by doing this kind of work of being able to alter the contents of memories, that it gives a kind of blueprint for or a kind of sketch from the tricycle of how the Lamborghini might work. So for instance, one of the findings that we had was on being able to artificially activate a positive memory, but while the animal was experiencing something fearful. And that actually interfered with the fearful states that the animal would acquire. In humans, the beauty of it, right, is that we don't have to put optic fibers and genetic fanciness in your brain. I can ask you, like, what memory do you hold near and dear that means something to you? So that already is memory recall by just asking, by prompting you with a question, and it's as non-invasive as it gets. So that's all to say that the goal is now asking like whether or not we can showcase like the power of positive memories by trying to actively recollect them during a modifiable period when certain memories, especially those linked to some kind of trauma, are particularly modifiable. Or we can restructure, in a sense, what that negative experience means by trying to disentangle the emotional components from the actual event that happened, and then that takes us through different corners of the brain as well. And that's the goal, is that can we use what we did in mice as the blueprint of how this may work in humans, and ideally in as non-invasive of a way, with either targeted modulation or something with intentionally sitting with a positive memory at the right times to see if that can have any therapeutic purpose. Yeah.
Hi, first of all, thank you. It was so fascinating, the entire story and the way you told it. So
Thank you.
I bow to you. Thank
Thank you.
My question is the dance between the memoir, if you will, and the science. And I'm just curious, your process, did that channel through you? Or how did the process of weaving the two and prioritizing what over what, how did that work for you?
That was the hardest part of writing this book. I think of it as like when it came to writing or talking about the science, it felt like, just to use an analogy, I'm right-handed. It felt like I was just being asked to throw a baseball with my right hand. And when I talk about science, I'm like, I can do that. I can do that. I enjoy it. I have a bunch of internal frameworks for being able to conceptualize science in a way that's accessible. It's why I love teaching and why I love connecting with audiences. The memoir part felt like I was trying to pitch with my left hand, where suddenly I was like, oh my God, I stink at this. And this is actually like, I don't even know how to aim properly. And that actually took, I make the joke with my book agent now where I'm like, that's the reason why this thing took almost a decade and that I promise the next one will not take a decade, because I learned how to pitch with my left hand as well. That to me was the hardest part, because I realized through life, friends, family, therapy, all of the above, that there's so many thoughts and feelings that I had that I could maybe convey in like somewhat of a understandable or intelligible way, but I needed to practice putting those thoughts and feelings into words again and again until I was like, that sentence gets at the feeling I was trying to convey, or that paragraph gets the overall sentiment that I felt the day that shoe passed, or things like that. So the memoir part was in and of itself very literally therapeutic, because I learned to like put all of the cacophony in a brain about feelings and emotions surrounding pretty heavier topics into words, and then into words that can be a story. So that was the hardest, but admittedly the most rewarding part, because in like a kind of cheesy but not cheesy sort of way, I learned so much about myself and my relationship with my own memories through that process of putting those feelings and thoughts into the written word and into a story. It was pretty amazing. Like that felt like I was unpacking different parts of me that I didn't know how to self-express before. And like some of us do it through music, or some of us do it through sport or anything, and then writing was the way that I channeled that into stuff that hopefully made sense. Yeah, so difficult, but the most rewarding part.
Awesome talk.
Thank you.
And very good storytelling. My question is about memory, but specifically if we can impact traumatic memories that occur during infancy, so before, let's say the age of 1, where you can't recall, but your body still has some kind of response. Does it only work with memories that we can actually recall, or does it work with any memory that's stored?
If it's in rodents so far, the work that's come out, which is hopefully a sneak preview of the kind of stuff that one day will be medically possible in humans in a medical setting. In rodents, at least, it's pretty amazing, because there's like a dozen studies now that have been able to find the cells that hold on to an experience that happens at infancy, in mice in this case. And then the animals, they show amnesia just like humans later on in life. They don't show any evidence of recalling that memory. But if we go in and stimulate those cells, we suddenly pull that memory out of amnesia and bring it back at the forefront of the animal's mind. And then that, when it's now active, not only is that kind of amazing that you can pull an infantile memory out of amnesia, but then while it's active, you can do stuff to it. You can enhance it or try to permanently suppress its like emotional components, for instance, or just let it be and have it go restored back into the brain and back into an amnestic state. I think that the kind of common theme here is that memories seem to be pretty hard to break, in the sense that we may store more memories than we think in the brain, and we simply just don't have access to a ton of these experiences, and infantile amnesia is the one amnesia we all share. But there's some evidence in humans, if I remember correctly, showing that like if you can somehow miraculously get an infant under an MRI, you do see some like semblance of hippocampal activity during episodes and during certain experiences. So there's every reason to believe that experience is it's leaving an imprint on the infant brain. but it's probably, as you suggested later on, more felt than recalled. How in humans, up in the air, like still no idea, but at least in rodents that we know that we can pull that memory out of amnesia and then start tinkering with it. So you can imagine a case of an aversive memory where we can pull it out, bring the feelings and the episodes at the forefront of the mind, and maybe try to therapeutically dial down the more negative or traumatic emotional components of it. In rodents, I think that that's fully possible. The hope is that that would be possible in humans in a therapeutic way as well.
I just felt compelled to respond to the person who said that he didn't have any memories from his first year. I have 2 memories from my first year, an increasing number of memories for each year after that. I don't know why, but they're there, and I just wanted you to comment on that.
Yeah, there's in both rodents and humans, there's this phenomenon of memory linking that, you know, if something amazing happens, you're likely to piggyback the things that happened before and the things that happened afterwards. Or if something horrible happens, you're likely to import the memories that came before and that came afterwards with that particularly salient event. It could be that these memories are chained together by some kind of cognitive common denominator. That could be emotion, for instance. Or it might be something else that we simply don't know. Like I don't know if there's a common pattern to the kinds of memories that are chained together, but at least the ones that have been studied in the lab tend to have some emotional component that ties together what happened before the event as well as what happened afterwards. And this can go across days and weeks. Like it's not necessarily limited to like what I was doing before this and what I'm doing after this. It could be that our brain is then finding the common denominator between the experience of right now and a subsequent experience that I might have in a lecture hall when I'm talking about this book that then becomes linked with this, and so on and so forth. So I would posit that those memories have some kind of like unifying theme to them, but that's a total speculation.
I don't think they do, actually.
Oh, great. That means the brain's even more complicated.
I'll think about them again when I get home and try to see if that's the case.
Yeah, thank you.
All right, and I think this will be our last question before we go over to the library.
At the beginning of your presentation, you spoke about the easiest way to prove your theory was to focus on fear. What would it be if you actually wanted to treat someone that has addiction, which is associating very good feelings to that thing, like alcohol or whatever? How could you then, would that work reversed and then put fear in those people, or is it not ethically probable? I don't know, like, what do you think about that?
Yeah, there's a couple of ways, like with addiction in particular. Like we had a couple of studies a few years ago that showed that if we find the cells that were active when an animal was experiencing ethanol exposure, if we reactivate those cells again and again and again and again, it seemed to suppress the animal's like wanting to be exposed to more ethanol. So it's kind of like exposure therapy, but inside of the brain. The real way of doing this, I think, is by saying we have to figure out what our triggers are in life, and then what it is about, for example, it could be a beer bottle, it could be a syringe, it could be a powder, but there's very real visual and smell and auditory, all sorts of triggers that we have. And if we could find those and either do something like exposure therapy or be exposed to them and then tie some negative stimulus to it. With drinking, there's a couple of examples of taking like some drugs that make you actually get sick if you drink, and that's a really potent way of getting someone to not want to drink because you're just going to essentially get instant food poisoning afterwards. And that works because you're, in a sense, reassociating what that trigger is to something negative so that you won't want it anymore. The caveat of this is that I think it's very individual specific, that for some people that'll work really well, for others it might actually even make things worse, and that's where it gets a little tricky because everyone has their own secret sauce that may or may not work. So something that's a combination of either exposure therapy or associating a trigger with a negative event and so on can be all effective, but it depends on the person. In mice, super possible. In people, that's where that variability I think becomes a lot more difficult to address, right?
All right, well, I'll just ask everyone, please, if you'd like, come over across the hall to the library, make some good memories. Steve will be over there signing books and having some good conversation. Let's thank him again.
Thank you all so much.